Hi. I know it's a challenging hour, but just a little bit more and then we'll have a coffee break. My name is Naama, and today I'll be talking to you about quantum energy dynamics in photosynthesis and how it can give us a fresh perspective using noise analysis on these interactions. Now, photosynthesis, as we know, is the engine of life on Earth, converting sunlight, carbon dioxide, and water into oxygen and sugars that we animals can use. The interesting thing here is that it happens at extremely efficient rates, especially for the initial capture of photon and conversion to electron excitation. Now, simply put, photosynthesis is the conversion of light energy to chemical energy, but it also has byproducts, heat and fluorescence. Now, fluorescence is especially useful for us because it can provide a non-invasive probe to monitor the energy dynamics within the organisms. Now, for me, in photosynthesis, the questions that interest me are always biological. Like, how do photosynthetic organisms cope with changing conditions? For instance, light energy is not constant. We can think about the day and the night, but also in the ocean water column, where many photosynthetic organisms reside, the light energy attenuates with depth, and also the wavelengths change. Even on the forest floor, which we think of a calming and quiet experience, at the level of a single leaf, it can transfer between very low levels of light in the shade at once, and then quickly into high levels of light in under full exposure. So the challenge for photosynthetic organisms is how to maximize efficiency while minimizing damage from excess energy. So to try and answer this question, we wrote a review paper a few years back, looking at photosynthesis on the quantum classical border. To paraphrase an example by Rink van Gondel, you can think of the energy transfer between pigments as a football field filled with holes, where you have different energetic excitations as balls moving around that field. You have small balls, which are small enough to fall into the holes and become localized excitons. And then you have larger ones that just don't fall in, and then they roam around the whole football field, and they are delocalized excited states. In this paper, we explored the flexible switch between an ordered state, where we have delocalization of energetic excitation and efficient energy transfer, versus the disordered state, where we have localization and therefore quenching of excess energy. Now, I'd like you to remember this slide because it gives context to the rest of the talk. We looked at examples of disorder and order switching. First, in vitro, Ida Eisenberg and colleagues took extractions of pigments from photosynthetic organisms and saw that when you dried them, they create these beautiful self-ordered states that are very ordered. Now, when you look at the lifetime of the excitation, as the pigments dry, the lifetime reduces, which means that in this ordered state, we have higher coupling and a delocalized exciton. Moving to an in vivo example, we looked at desert crust cyanobacteria. Now, these are keystone species because they stabilize the ground surface and make it possible for higher level plants and trees to come and colonize the area later. Now, living in the desert comes with extreme changes in temperature and hydration, the most significant being in the morning. During the dawn, there's a very short space of time where these organisms both dry out and are exposed to much higher levels of light. So how do they cope with that? Well, Liat Barreal and colleagues saw that when you look at transmission electron microscope images of these cyanobacteria, in the wet state, they are very ordered photosynthetic membranes, while in the dry state, they are disordered and scrambled. They also saw that at the same time, in the wet state, there is efficient energy transfer, while in the dry state, there is a quenching of excess energy. So these cyanobacteria utilize the fact that they're drying out and at the same time, they're quenching the excess energy from the sunlight. So we talked about the ordered versus the disordered state and the flexible switch between the two. We looked at two examples, pigment nanowires and desert crust cyanobacteria. But these were measured using conventional methods in photosynthetic research. But now I'd like to talk to you about unconventional methods, and that is my current research, moving from the conventional to the unconventional. And specifically, I'd like to introduce you to the concept of quantum versus classical noise in biology. But first, what is noise? Perhaps, like me, you're thinking of something like this. But I'm not talking about acoustic noise. What I am talking about is signal fluctuations. When you look at any signal over time, there will be a trend, but the signal will fluctuate around this trend. Now, when you subtract the trend or the average, you get the noise, the deviation from that average. What can you do with this? Well, we do two main things. The first is to create a histogram of the noise, which is what happens when you go along the signal and count the number of specific deviations from that average. This becomes the x-axis on the histogram, and the y is the amount of times it happened. The other thing you could do is a Fourier transform to look at the spectrum of the noise using a parallel spectral density graph. But why is this even interesting? Well, to begin with, it's not commonly used in biology, and that's a shame because noise can be super informative. If we look at the next level of information beyond the trend, we can look at a whole new range of possibilities. For instance, we can ask, what kind of noise are we looking at? So in general, most classical noise is Gaussian in nature in the histogram. But in quantum systems, where the energy levels are quantized and distinct, we expect to see multiple Gaussians around distinct energy levels, which leads us to a more complicated histogram. In our first project, in collaboration with Naama, a fellow PhD student in our lab, and Professor Gregor from Ben-Gurion University, we looked at the question, can we use noise to identify biological versus chemical samples? A very simple first step. For our experimental setup, we have a laser input modulated by a chopper hitting our sample. The sample gives off fluorescence, which we pass through a high-pass filter, and we put both of them through a lock-in into our spectrum analyzer to see our results. Our samples are first living cells, so we have both marine and freshwater cyanobacteria. Then, from those same cyanobacteria, we extract the chlorophyll pigment, which we're interested in, using acetone. Now, this acetone is quite liquid in nature, so to further compare, we also add to a different sample a polyethylene glycol to that acetone to create a more viscous solution. So now we're looking at the same pigment, but in different environments. We also have a reference, which is just an empty cuvette. Now, the first result we saw is that for the pigment in solution in the liquid solution of acetone, we have a wide and not uncomplex histogram with multiple peaks. This is quantum-like. Secondly, when you compare it to the other pigments, we see that for the acetone, the histogram is wider. So you can see that both for the cells and for the pigment in the viscous solution, we have a single peaked, narrow histogram. What we think is happening here, that the pigment in solution in the acetone has access to higher degrees of freedom, translation, rotation, vibration, and these show up as extra peaks in the histogram. In contrast, in vivo, pigments are restricted in cell membranes, much like the pigment is restricted in the PEG solution. Now, this ties in to the ordered versus disordered state we were talking about before. But we wanted to understand this a little bit further. And to see that, we need to look Now, what happens to chlorophyll during that excitation process? So what you need to know is that chlorophyll can be excited to one of two states. The first is singlet chlorophyll, and that's what we want, because that can lead to chemical energy along the line. The second state is triplet chlorophyll, which is what we don't want, because it can interact with triplet oxygen, leading to singlet oxygen and reactive oxygen species, which we were introduced to just 2 talks before. These can be very harmful to proteins and even DNA in the organisms. So generally, we'd want to avoid. So we wanted to see what happens when we take oxygen out of the picture. To do that, we simply bubbled nitrogen into the existing acetone pigment samples. What we got was that oxygen depletion narrows the noise histogram, and now it's comparable to the polyethylene glycol, the more viscous solution histogram. What we think, therefore, is happening is that biological membranes may restrict the formation of triplet chlorophyll using a protective mechanism of ordered state. Now, to answer the question of why this is interesting, well, first we're exploring complex, perhaps even quantum phenomena in photosynthesis in a whole new way. Secondly, noise allows us to map the energetic landscape. And what do I mean by that? Usually, we're looking at the overall trend, at the dominant process happening inside the photosynthetic organism at that time. But using noise, we can look at the whole landscape of opportunities available to those pigments. Now, taking this further, I'd like to show you two more studies. The first looking at plant dehydration dynamics over time, and the second is a long-term study of coral ecology. So looking at plant dehydration, we're using much the same setup, except that now we're looking at plants. And what we saw is, first of all, when you compare the wet and the dry states, you can straight away see that in the wet state, we have these ordered thylakoid membrane stacks, and in the dry state, they start to unravel. As we were talking, the ordered versus the disordered state. How does this translate into noise? Well, we see that in the histogram, for the dry state, it's much wider. Okay, so the disarray leads to a wider range of possibilities. And furthermore, in the power spectral density, or the Fourier transform of the noise, we can see that activity is depleted across all spectrums, and the PSD goes down. And we take that to mean that we have overall less activity in the dry state, which just makes sense. Now, I won't show you here, but when you compare this to classical and well-accepted measures of photosynthetic performance in plants, we can see that our measure is more sensitive and better differentiates between dry and wet states. The second project, looking at the coral monitoring system in Eilat, created and managed by Professor Maroudas-Sklare. Here we're looking at 3 different locations with 8 coral colonies each of different species over 4 years' time. Now, this huge database lets us ask really interesting questions about an ecological keystone species that, as we all know, is critically endangered by climate change. Now, the first and most striking result that we saw here is that when you compare the full width half maximum of the histogram, you can see that during the day, it's actually narrower than during the night. Now, this is consistent. We can see that each different dot here on the plot is a different coral colony at a different date period throughout the year, meaning that for different species, different depths, go to the shallow and mesophotic here, and for different seasons, we consistently get higher noise at night. Now, this really surprised us, okay? We expected that during the day, there would be more photosynthetic activity and therefore we'd get more noise, but we got the exact opposite. So looking more closely at the data, we tried to understand what was happening. And what we saw is that during the night, the corals exhibit higher sensitivity to artificial light induction, which is related to classical photosynthetic measurements that we observed during that time. And this means that this may have a connection to light pollution, which you can think of the implications for this critically endangered species. So, to summarize, we started talking about photosynthesis and the question of how photosynthetic organisms cope with changing conditions. We talked about the ordered versus the disordered state, related to efficient energy transfer and the quenching of excess energy and the flexible switch between the two. We looked at conventional methods, both with pigment nanowires and desert crust cyanobacteria. And then we looked at unconventional methods, looked at noise analysis in photosynthesis, first with the in vivo versus the in vitro example of differentiating between chemical and biological states and seeing the complex triplet-singlet dynamics that we were sensitive to. Then we look at plant dehydration dynamics. And finally, coral ecology. With that, I'd like to thank my wonderful friends and colleagues at the Paltiel Q&E and at the Keren Labs, and leave you with a take-home message that noise analysis is a powerful new tool for investigating energetic dynamics in biology. Please feel free to keep in touch and scan the articles if you're interested. Thank you very much for listening.